So I think it's time. I would like to start now. I'm Cherry Kubota sitting in Columbus, Ohio, Ohio State University. Welcome to Vegetable Grafting webinar. Um, this is the first one this year, but we have been doing a few months now. Actually, this is number 3, and then we will be doing monthly basis. And I will introduce today's speaker soon, but he's a professor in North Carolina State University and our project leader, Frank Louws. But let me quickly introduce you what you expect for the upcoming webinars. Again, this is a monthly webinar series organized by Vegetable Grafting Project Team funded by USDA Specialty Crop Grants. And after today's, there are 4 webinars already scheduled. The first one after this is by researchers in University of California Cooperative Extension, Brenna Agadar and Jim Miao. And then the March webinar is by Richard Hassel, Clemson University. And April webinar will be by Tony Kainas, Clemson University. And May webinar will be by Carrie River, Kansas State University. And more webinars are going to be scheduled monthly basis. And you can find the update on this Vegetable Grafting org website. So as a housekeeping, you want to keep your microphone muted. And then if you have questions, you're welcome to use microphone because that's easier for us rather than reading through the chat. But you can also use chat area as you wish. So that's the sort of way to do this webinar. Hopefully we have ample time to get questions and answers at the end. One more announcement, we will be organizing International Vegetable Grafting Symposium. This is the second one as an international symposium focusing on vegetable grafting, science and technologies, July 14th to 18th, North Carolina. Currently, we are accepting abstracts. If you are interested in presenting your research outcome, and then also accepting those who are interested in a trade exhibition, we will have that opportunity in that symposium too. So it's a great network, learn what's going on, not just US, but other countries too. With that, I would like to introduce today's speaker, Dr. Frank Louws. And while I'm introducing him, I would like to ask Frank to share your screen. So Dr. Frank Louws is professor and the department head of horticulture, North Carolina State University. Up until recently, he has been director of, I have to look at the Center for
Integrated Pest Management.
Integrated Pest Management. Sorry, my cheating is gone somewhere. And then he has been the project director of this USDA SCRI vegetable grafting grant program, leading 10, probably 11 now, 10 institutions consisting or involving 38, 39 researchers. It's a great group, very interdisciplinary, and he's a great leader. So he's going to talk about use of vegetable grafting for soil-borne disease management. Okay, so all yours, Frank.
All right, and we're recording?
Yes, it's
excellent. Okay.
Recording going.
Yeah, well, welcome everybody, and it's a real privilege to be here and to present this information. As Sherry said, I've done a lot of work in plant pathology, but now in the Department of Horticulture, And it's a great department. And of course, a lot of the work that we do has always been on horticultural crops. And so I'm really pleased to be part of that department. What I'd like to do is, Sherry introduced the project, and I'd just like to give a little bit of an overview of the project just to remind everybody what it's about. And I'm in a room with a big screen, and my camera is a bit low, so now and then you'll see me looking up, and I'll lose eye contact with the camera. But as Sherry indicated, it's a multi-institutional transdisciplinary program, and what we really enjoy. And Sherry, can you see my pointer?
Yes.
Excellent. Okay. What we've really enjoyed is that we've had so many private partners, seed companies, robotic companies, grafting nurseries, grower associations, and of course, a lot of growers and other stakeholders are involved. The core of the project has multi-components, but it's really built around fruiting vegetables, the Solanaceae and Cucurbitaceae. And our desire is to ask the question, where will grafting help the most in field production systems for tomatoes and solanaceous crops, peppers, etc.? And where will grafting help the most for cucurbits? And so we have working group leaders for each component, and that's the names. This is Shin Sao from Florida, Richard Hassel from Clemson, Penny Perkins from NC State, just post-harvest. And then we asked the question, Dilip Panthy is here in the room with me. It's the breeder here, and also David Francis and others. We asked the question, to what extent can we advance rootstock genetics to optimize rootstock performance? And then we have a technology-based group, and Cherry Kubota leads that. And of course, each one of these has a large team of people. But this really asks the question, how can we optimize grafting technologies for the grafting itself? How can we optimize transport? How can we optimize plant physiology? How can we optimize cost efficiency? And then we also do social and economic analysis on all the systems, and Dr. Gao from Florida leads that. And then we have a very strong extension outreach component. Dr. Kleinhentz from Ohio State University leads that. So just an overview, done a lot of work all across the states on fruiting vegetables, and as Cherry Kubota pointed out, you can see the outcomes of these projects at vegetablegrafting.org. That's an excellent site where we try to post everything we can. I've really enjoyed this project, and the whole team in the U.S. has, because adopting grafting into the U.S. field production systems is, with this level of effort, is more of a new venture for us. And so we've really leaned heavily into international collaborators. And so we're really pleased for the international collaborators and people on this conference call. And it really takes an international body of knowledge to go from baby steps to running. And that's, so we've really leaned into the international opportunities. These are shots from Italy at a conference, China, and the Vegetable World Center, the World Vegetable Center in Taiwan. Dr. Kubota invited you to the International Symposium, so I won't spend time there. You know, it's a great opportunity for us all to get together, and the registration's open, abstracts are open, etc. All right, so, you know, I think one of the main challenges that we face with vegetable production systems are vegetable, you know, biotic stress, which of course is diseases, and that's what I'm going to be focusing on. But there's also the whole world of abiotic stress, drought, salt, temperature tolerance. And then, you know, we like to ask questions, well, how can we optimize production systems by breeding for roots and using rootstocks? You know, we want to optimize our resources, water, nutrients, land resources, decrease environmental impact, enhance fruit quality, enhance yield, and all kinds of other benefits for grafting. But of course, today we're focusing on diseases and disease management. The challenge, I think, with vegetable grafting, or with soil-borne diseases in vegetable systems, are a bunch of things. And all over the world, we see more and more intensification of production, less rotation. So we see more and more pathogen, heavier pressure from pathogens, the inoculum pressure. And then a lot of our growers want to grow unique plants like heirlooms, and typically they don't have good genes for resistance. And so to meet market demands, many of our growers and stakeholders are growing susceptible cultivars. And then we live in a global environment, and we see a lot of movement of global produce, and so we see movement of the pathogens also, novel pathogens, invasive species, etc. We also see a high interest in alternative production systems, organic high tunnel systems. So there's less tools available like fumigants, etc. And so, we need mechanisms to control soil-borne diseases. AVRDC, the World Vegetable Center, of course, works a lot with resource-limited farmers, as do a lot of members of our team through various international programs. And then we're seeing loss of products that offer disease control, such as the fumigant. So we have a major challenge in managing soil-borne diseases, and I always like to advocate for an IPM approach. We want to know the pathogens, we want to diagnose them properly. To what extent can we eradicate them? To what extent can we use products, biological control products or other chemical fungicides? To what extent can we learn more about who the enemy is, their dynamics and their persistence, and how they interact with plants? And then, of course, what role does host resistance play? And then, the big question I think often is how does this all fit into farming systems approaches? And I won't talk about that, but we really want to think through about how to optimize soil health and farming system-based approaches to solving the soil-borne problems. The challenge with plant pathology and diseases is this shows a single cell of a plant. And of course, we have mycelium from the fungus. We have a head of a nematode. We have a bacterium, 10 millionths of a meter long, maybe. And so a lot of times our enemies are not visible. And that presents a challenge to a lot of our stakeholders. Of course, we see symptoms. This is root knot on tomato. We see signs. This is the female nematode that was in the root in the gall. The other challenge we face with soil-borne diseases is we have few thresholds for action. And we have assays to assess nematode populations, and those are pretty routine. We have assays to assess for verticillium, inoculum pressure. Those are not always routine. Some places specialize in that, but it's not like we have labs in the US or other parts of the world that routinely do that. And so most growers go by land history. What has been their problem before? So in most cases, since we can't have thresholds, we need a proactive approach to managing soil-borne diseases. In the past, that's been methyl bromide. It's been a major fumigant or other fumigants. And people all over the world have looked at other fumigants. I call that tactic substitution. People have looked at non-fumigant approaches. I call that tactic diversification. And that's really where genetic resistance comes in. We're looking for other ways to control diseases. And then, we want to do tactic development. How can we advance microbiome, microbial ecology, farming systems research? That's really at the forefront of research now. And then this one here, I love it. I hope there's a lot of students here. This is your future. How do you design sustainable systems for soil-borne disease management? And the science is really exciting right now. It's a great time to be a student. So the challenge has been in terms of genetics is that we might have host genetics that confers resistance, that gives resistance to different pathogens. The problem is a lot of time that resistance is not durable. It might last for a while, or a new species comes in, or a new pathogen, and then that cultivar fails. And the other thing is a lot of the genetics that gives good resistance are not incorporated into lines that have commercial values for stakeholders. And so, that's what grafting does. This is a slide Terry Kubota had put together, and one of the drivers for grafting in the 1920s and 30s in Japan and Korea was to manage fusarium wilt of watermelon. And so it's not new technology, and in fact there's literature as early as the 500s in China. And here in the US, people have been using certain Jimson's weed rootstock for root knot management. And so the role of grafting for soil-borne diseases has been a very important tool for many years. Just to remind everybody, what the advantage, of course, is we have the scion, we have the rootstock, and the advantage is we can breed for rootstock and or select rootstock for the problems that we have within the field. So local selection of rootstocks for local problems. And then we can still grow the scion, and I call it, in this case, this is a tomato. I call it YFT. This is your favorite tomato. You can put it on almost on any rootstock and get the tomato that gives you market value and the rootstock that might give you disease resistance. In North Carolina, my joke is, and my growers don't like this, of course, my joke is that we're really blessed because we have mountains in the west, which is a temperate climate, and we have subtropical conditions in the very southeast. So we have a high level of geographic diversity, which means we get most diseases that occur on tomatoes. So that's good if you want to work on it. It's not good if you want to grow tomatoes. But it just shows, you know, we have the temperate pathogens like verticillium. They don't occur in the eastern North Carolina, but they're a significant part of our systems in the mountains. And grafting is a very excellent tool for what we call race 1, and it's a very modest to low efficacy tool in race 2. And then we have the fusarium wilts, race 0, 1, and 2, and I'll talk more about this. But again, grafting has high potential to give fusarium control. Bacterial wilt, Rostonia solanacearum, we have race 1 here, and I'll show a lot of work of how that's been helpful. We've had a few surprises, Sclerotium rollsii, southern stem blight, and I'll talk about that. What's really been exciting is we're seeing in watermelons, in peppers, some phytophthora root and crown rot control, and of course, root knot control. And so, in North Carolina, we've been able to look at a lot of these pathogens because we have the geographic diversity. So what I want to do is just remind everybody what we mean by races and host resistance and gene for gene sort of models. We have pathogens and their shifty enemies. And as many of you know, the fusarium wilts of watermelon, fusarium wilt of tomato, they're very host specific. And so the tomato one will never cause disease in watermelon and vice versa. And there's a lot of Forma specialis that cause unique diseases on specific plants. And it's so specific that certain lines of watermelon will give resistance to certain races. So we have genetic races of the pathogen in the international nomenclature, let's say for tomato is 0, 1, and 2. In the US, we often call them 1, 2, and 3, race 3. We also have closely related species like Fusarium oxysporum, Forma specialis, Radicis lycoprisci. It's a crown rotter, not a wilter. They're closely related, but they're very different diseases. So if you have a pathogen, let's say for watermelon or tomato, we call them race 0, 1, 2. And the only reason we know they have races, we have these races, is because we have genes that can differentiate them. So there might be more races out there that we're not aware of. But the concept is we might breed a tomato line and we get resistance to this one. But then we get mutations or we get migration of new invasive races. And then all of a sudden this host is susceptible to race 1. So we have a breeding program, we develop a resistant line, and it gives resistance to both of these races. And then we come up with the pathogen, a new pathogen comes in, or we get a mutation, and then this gene is no longer effective, and so we discover a new gene and we incorporate that. But the problem is when we use, let's say if we use tomatoes and tomato breeding programs, or watermelon and watermelon breeding programs, you know, the danger is there might always be another race that can come and overcome the resistance that we have. One of the powers of grafting is that we can use non-host resistance. So like I said, a tomato strain will never go to a watermelon strain. And so if you can find tomato rootstocks or watermelon rootstocks from a different genus or species, you can have complete resistance against all these host-specific pathogens. So I just wanted to review the concepts of races, differential hosts to tell those races, and then host and non-host concepts. So we're just all on the same page. So I'm going to talk first about bacterial wilt. It's a, you know, like I said, it tends to be a subtropical pathogen. Although we do get it in the mountains, we call it southern bacterial wilt. The pathogen has high genetic diversity. It gets into the plumbing system of the plant and clogs up the xylem tissue. And so we see a rapid wilt of the plants. And once in the soil, it can persist for years and years and years. It has a wide host range, and you can't fumigate against it. So there's a lot of challenges with this pathogen. The other challenge has been when you breed for resistance, good resistance tends to be tightly linked to poor fruit size. And so it's been hard to develop cultivars with high levels of resistance. So this is some of our earlier work. This shows the percent of disease, 80% plant death versus 0. And these are some early screening trials we did, just looking at, here's our control, Florida 47 we call it. Here's Maxifort rootstock. It has no bacterial wilt resistance. Excellent rootstock, but not for bacterial wilt. And here's the line called Chonggang from Seminis. Here's the breeding lines, Hawaii 7997, 798, D. Palmer 106, CRA 66, an open pollinated line, giving excellent, excellent control of bacterial wilt, 0 to 10% plant wilting. And when you look at yield, of course, the control, Maxifort, very low yields, maybe 15 tons per hectare. And then our superior rootstocks, we're getting about 65 tons per hectare. And even this open pollinated one, it tends to do fairly well. It gives good control and also gives good yield. So if you're an organic grower, you might be able to grow your own seed for rootstocks. Hawaii 7997, 7998, excellent, excellent resistance to bacterial wilt, but they don't give good yields typically as a rootstock. This is a picture from that field. It's a bit fuzzy on my screen, but I have it on, but it just shows you the question is, it's a philosophical question almost, to graft or not to graft. You see here in this farmer's field, we have pretty well 100% wilt versus 100% plant stand. So really with grafted rootstock, we're putting the best land back into tomato production by grafting. We do a lot of on-farm research. This is an example. This grower grows about 101 acres of tomatoes, hectares of tomatoes, and we have such great cooperators. This is a randomized complete block design trial. We have very large plots. We work with companies to provide the grafted plants. In this case, we had multiple treatments, and you know, they give us a crew. We work with that crew. Everything's color-coded. We put it all in. We collect all the data. So we'll come and we'll rate for bacterial wilt. You can see these plants here just going down, where some grafted plants are standing up well. And then the grower helps us harvest all the data, but just shows here in this on-farm trial, this is the control. We have about 40% plant wilt. These are some early lines. They were Reichswan, they were numbered lines. They gave us pretty well 100% control. And one of these lines is now called Shield. We did a lot of work where we pinched the plant, we let 2 leaders grow, we trained them in the row with the string, and we can reduce the plant density. And actually, in this particular on-farm trial, pinched plants gave us 14% higher yield per acre. But that's not always the case, and it's more costly to do a pinched plant. This is another trial in the mountains. This is more recent data, 2017. We also did in 2018, but it just shows high disease levels in our control. And here we've expanded our rootstock selections, and they're giving us excellent, excellent control. Xinxianggang from Seminus, Bowman from Cicada, RST 106 from D. Palmer, Armada from Taki, CRA 66 and our Hawaiian lines. Our Shield from Reich Swan has not been performing as well, so we don't recommend that anymore. And then, of course, this high level of disease control translates into high level of yield. Okay, but these are the ones that we're really comfortable recommending. The Armada Xinxianggang, which is Seminus, again, Cicadas, DP10, D. Palmer seed, and just giving us excellent, excellent yield compared to the control. You can see Shield here was modest, Hawaii 7998, modest. So we just see good disease control translating to high yield. All right, and so we have a very diverse state, like I said, and we've shown that those recommended rootstocks do really well throughout all parts of the state. We feel the population of our bacteria is slightly different genetically than the population here, although we haven't systematically proven that all. The other thing we've done a lot of work on is taking some of our best rootstocks and just ask the question, how do they perform in the absence of disease? This is Red Mountain and Picus, two tomato types, different rootstocks. And the long and short of it is, after quite a few years of work, is that typically the rootstocks give equal or similar yield, and sometimes we see better yield. So there's not a negative cost to grafting if you don't have disease pressure in that part of your field. The challenge is, there's many. We need to learn more and more about the genetic diversity of the bacteria, understand mechanisms of pathogenicity, understand microbial ecology, and then we want to be smart about figuring out who exactly is our enemy and what's the potential of threat. We also want to work on the tomato side using, like Dr. Panthy here has a program also to do QTL mapping. This is work from Taiwan. We want to get higher and higher resolution of these QTLs, and so we can smartly use these genes against the known threat to get optimized yields. And for students here, there's so many PhD questions here. It is so wonderful. This is a rather unique situation. This is southern stem blight. It's a fungal disease that persists in the soil for years. This was a serendipitous finding. We did a grafting project on an organic farm for Fusarium, and we ran into very heavy southern stem blight pressure. You can see if we don't graft, we have 80% plant death. This was not on the organic farm, so in this case we used a fumigant. It cut it back a bit. It was just telone, not chlorpicrin. But you see here, this rootstock gave almost 100% control. That's another PhD thesis. What is the mechanism of resistance here? But it was really enjoyable to see that data. Likewise with nematodes, this is a 12. This rating 0 to 12 is a scale we use, 100% galling. You can see if we don't fumigate, if we don't use rootstock, we get 100% galling. If we fumigate with a nematicide, 1,3-dichloropropene, we get good control. You can see here, if we use Maxifort, Beaufort, or Big Power, we significantly reduce the galling. In fact, Big Power, very interestingly, gave complete control. And this is in North Carolina in August. Those soils are extremely hot. We know the MI gene can fail when the temperatures get above 28 degrees Celsius. We see that turn out to yield. This is pounds per plot. Here's the control. Fumigation really helps. The grafting substantially improves yield. And then what was surprising is when we looked at the nematodes in the soil, we saw differences in the season, but by the end of the season, we had high nematode populations, except for this Big Power. which means one of two things. One, somehow it gives complete resistance against the nematode, or this MI gene in this genomic background was heat stable. And again, that's another great PhD question. What is the mechanism of this complete control? How can we move it into other rootstocks? Verticillium wilt, that's the nemesis for us in the mountains. Very aggressive disease. You can see the wilting occurring here. And the pathogen persists in the soils for years. We have good race 1 resistance, but most of our strains are what we call race 2. Race 1, this is the VE gene. This is several lines of the gene that we've sequenced just to confirm. The VE gene interacts with the effector of the pathogen, confers complete resistance, and that's race 1. But if you have a mutation in that VE gene, then the verticillium pathogen can overcome that. And or if we have a mutation in the effector gene, then we can have what we call race 2. Race 2 is everything that can overcome the race 1, the VE gene. So my analogy is often if I have a glass of milk, that would be race 1. All other drinks in the world are race 2. We don't know the genetics, but it's everything that's not race 1. This is some rotation work that we did, Kerry Rivard. This is on land that was grown in tomatoes for 12 years. And if you don't rotate and you don't graft, you have very poor plant growth. If you have crop rotation every 3 years, then you have a very good response. But if you graft Maxifort, and if you don't rotate, so this is 12 years of continuous tomato, or Maxifort on this as a rootstock, you get really good plant growth. And a lot of times we use Maxifort as a model rootstock. It's a bit expensive, but it really gives good vigor. And so functionally, we grew out of the verticillium pressure, which is the primary problem in this field. That was some early work. This is also some early work. Steve Groff in Pennsylvania grows a lot of his tomatoes in high tunnels in a continuous way. He often fumigates. You can see if you don't graft, this is his yield, a low yield. If he fumigates, he gets a distinct response. If we graft to Maxifort, if we fumigate or don't fumigate, we get a high yield response. And so after some work on his farm, he completely converted to grafted plants. This is field work in Western North Carolina, where we're looking at a lot of different types of rootstocks, some selections from Ohio, some selections from NC State, as well as all other available rootstocks. Some of these rootstocks come from different countries where they said they had race 2 resistance. But again, race 2 resistance in one country is not race 2 resistance in the mountains of North Carolina. What I wanted to show here is this is Red Defender, high level of disease. Even if we fumigate, this is non-farm trial. We feel this grower is using rates that are too low. But you can see here, we have Ohio lines and NC State selections that give very high tolerance against the pathogen. The Maxifort, when you graft it, Red Defender, it does not give a high tolerance to the disease. You get the same level of symptoms. So you don't get resistance to the symptoms. And I'll show you the yields in a minute. What's interesting is, let's say this CSU selection, if we use it as a rootstock, it's not that great. If it's the scion, then you get a very good response. And this is work that Israel has also done. Rootstock resistance to verticillium does not confer resistance to the scion. That's completely opposite and different than what we see with bacterial wilt. Rootstock resistance to bacterial wilt confers pretty well complete resistance to susceptible scion. Then this is yield. Look, disregard all the numbers, but what I wanted to show you is Maxifort gives such vigor. It's like a tolerance. We get the highest yields out of a rootstock that confers high tolerance. We don't believe it's a resistance. We haven't done the molecular work or GFP analysis, but also this NC State selection It's a selection that Randy Gardner has developed, also gives high yield as used as a rootstock. And so David Francis, Dilla Panthy, Randy Gardner are all using genetics, hopefully that will advance verticillium tolerance or resistance. This is just a quick slide I wanted to put in. We asked the question, we went into a field with no known disease pressure. We chose vigorous rootstocks, and this is 2 years in a row. This is 2018 data, so it's fresh off the press. You can see these rootstocks in the absence of disease pressure giving very good yield. We're getting 70 to 73,000 pounds per acre, whereas with the control, without disease, we have less yield, and shield is similar to the control in this case. The other thing that's emerged for us very recently, especially in some of our tunnels and greenhouse systems, is fusarium wilt. You can see the unilateral wilting here, the clogging of the xylem tissue, and then we can readily isolate the pathogen and work with it. And unfortunately, we did gene sequencing, looked at the diversity, and I was really surprised at this. We got race 1, race 2, and race 3 in the various systems. So that means for our growers, they got to choose rootstock that confers resistance to all 3 races. The challenge with watermelons is, again, fusarium wilt. We got 3 races. Then we have root knot nematodes caused by various melodogyne species. And then the big challenge is what happens if these 2 are together? So Tony Kyneth at Clemson and Richard Hassel have done a lot of excellent work on this. And of course, there's been a lot of excellent work all over the world. You see, if you have a rootstock susceptible, in this case, there was 80% wilt. And if you have the right rootstock, you virtually eliminate the amount of fusarium wilt. And this is against race 2. That translates into yield. In 2012, the control had 70% of the yield of some of the grafted plants. And in 2013, it was 40% of the yield. And this is published in Plant Disease. So Tony Kyneth makes a point that rootstocks that are not susceptible, the rootstocks are not susceptible to the host-specific strains. And so functionally, with watermelons, we have non-host resistance. For example, we have a completely different genus as a source of rootstock. And this rootstock so far has been resistant to all known races, and that liberates breeding programs to breed for roots in one program and breed for fruit-bearing cultivars in another. And Tony just makes the point that you got to make sure that the rootstock is compatible with watermelon scion. Sometimes you can have an exceptionally bitter flavor. The other challenge has been the fusarium root knot nematode. So Tony, and in a lot of systems, these work together in a synergistic way to create more severe problems. So Tony Kyneth asked that question, and he concludes that if you have rootstock, so here's the control. So this would be just the fusarium wilt and the fusarium with nematodes, but you don't get more fusarium wilt, and same with the rootstocks. We don't see an aggravated interaction. So if you have fusarium wilt resistance in the rootstock, and then you have nematodes, that fusarium wilt resistance holds up, which was a very nice piece of work. And that's in press. And likewise, this is really fun. Tony has a grad student, and they thought they would look at Pythium species. There's quite a range of Pythium species, and there's warm season species, there's hot season species, cold season species. But what was really cool, this is the control, 80% Pythium infection, and he could show that some of these rootstocks are conferring Pythium tolerance. Now, to my knowledge, that's the first case in the world for that in watermelons and watermelon rootstocks. But maybe there's other programs that have discovered that, but it's a really nice piece of data that the student has generated with Dr. Kyneth. The other thing, this is Josh Freeman, University of Florida, and the challenge has been most watermelon rootstock is susceptible to root knot. And Josh just highlights here, if you have a non-grafted plant, versus a grafted, you have substantially lower yield in the grafted plant because you have very, very high root knot problems on those grafted rootstocks. So USDA ARS as well as Richard Hassel have been breeding for root knot resistance in watermelon rootstocks. And this just shows carnivore rootstock, very, very galled. You can see the picture here, the level of galling. But look at this. This is called Bulldog. It's got a different name now. Excellent, excellent galling resistance. And what's really cool is, like that tomato, it pretty well zeroes out the reproduction of the pathogen, so there's no pathogen in the soil. So it's functionally a rotation to a non-susceptible crop, a non-host. And so Josh Freeman just makes the point that there's several rootstocks that have good root knot resistance. And so it's really important to have rootstock that has fusarium wilt as well as nematode resistance where both pathogens are present. Richard Hassel has done a lot of work with that rootstock. And this is Richard harvesting watermelons. You see, looks like they're having a little bit of fun. This is 2018 data. This is in the absence of disease. That line is called Strongback. That has that genetics. You can see he here got very good yields in total fruit weight compared to non-grafted rootstock. And it shows that it doesn't affect the number of fruit in this case, but increases the size of each fruit, thereby giving total higher yields. So this is now called Carolina Strongback. It's developed by USDA, Pat Wechter and others, Richard Hassel. It's got resistance to rates 1 and 2, got good root knot resistance, and Syngenta will be marketing it as Carolina Strongback. We're really excited about this because you need both parts of the equation. You can see grafted versus non-grafted here. Then closing down here, we've done a lot of work in North Carolina as well as many places in the world with Phytophthora capsicii. That is a waterborne mold. It gets into the roots, it crawls up into the crown, causes severe crown rot. You can see here zoomed in, you get complete crown rot, the plants collapse, the pathogen sporulates, and then it aerial disperses on the leaves and the fruit. And this is a field in North Carolina, 60 acres of processing tomatoes completely wiped out after a hurricane and spreading of the Phytophthora capsicii pathogen. This is 30 acres of plastic culture. All these beds have drip lines. They've been fumigated. 100% plant death on peppers. And you can see the pathogen on the fruit sporulating, the pathogen on the leaves. So if you don't control that crown rot, you get this rapid aerial dispersal, just like late blight of tomato. So this is really fun data. Again, this comes out of the SCRI work. Dr. Shaker at USDA ARS and Richard Hassel, as well as others. They have some breeding lines, and it just shows here these breeding lines give almost complete control of pepper, crown, and root rot. 100% plant death, nearly 100% plant control. And this is also in a field. You see we're getting complete control versus complete plant death in field work. Paladin has been our grower favored line with the best resistance we have for commercial lines in commercial pepper systems. This just shows a disease progress curve, non-grafted plants, grafted plants, almost zeroing out that problem. Finally, this is work in Washington, Dr. Carol Miles, where they have a lot of verticillium wilt pressure in watermelon. And this is some early work looking at disease severity, non-grafted plants up to 45% plant wilt, and then some rootstocks that are giving excellent control of that verticillium pathogen. And just, again, to graft or not to graft, this is a picture from Carol Miles. So finally, I just want to circle back on some of the challenges I think we face. First of all, we talked about shifty enemies, in that if we deploy certain types of resistance, we see new pathogen races come up. So we got to think smartly about how we use resistance in our rootstocks to get durable resistance. Can we pyramid them? Can we get multiple QTLs in there, etc.? Then, the other thing is a little tricky, and we don't think about this enough. Let's say if we constantly use bottle gourd rootstock. Well, in Japan, where they constantly use that rootstock, lo and behold, a new pathogen emerges that specifically causes wilt on that rootstock. It won't hit watermelon maybe, but now it hits that rootstock. So over-reliance on a single rootstock can lead to future threats. And the other thing is over-reliance on rootstock as a sole IPM tool can lead to problems. This was what happened in Italy. Growers went to grafting. They used the same rootstock over and over again, didn't use other IPM measures, and all of a sudden they got trouble with black dot caused by Colototricum coccoides, also like a crown rot caused by Rhizoctonia. So these pathogens, which we normally call minor pathogens, and they usually aren't an industry problem, when we're constantly using one method of control, we see other pathogens emerge as a problem. So we constantly need a holistic disease control program. We also have the challenge that you kind of triple your negative consequences when you graft, because you can have seed-borne pathogens on the rootstock, you have seed-borne pathogens on the scion, and then when you do all that cutting, you mechanically transmit them. This has been a very big issue for Clavibacter, of course, on tomato, Acetovorax on watermelons, as well as viruses on both crops. I think there's some really great opportunities. We need to learn more about the pathogens. We need to learn more about the genetics and bring that knowledge together to get durable disease control. The other thing, that's really cool is the question, to what extent can you breed rootstocks to change the scion attributes? So we might be able to change fruit quality, or there's even good data that some rootstocks confers resistance to foliar diseases like powdery mildew. So as we get more and more into the grafting and the molecular mechanisms of crosstalk between the scion and the rootstock, I think we can breed for more and more traits that have indirect effects. And then, the question is, can you breed rootstocks for microbiomes? You want to have your favorite microbiome, and maybe we can have the right type of genetics to favor certain microbiomes to suppress disease. So these are all fun questions. Sherry, this is your slide that you got at an expo, I think, in South America or something, and just shows, wow, what a great difference between, a conventional root versus some of the roots we have, and hopefully we still have time for questions.
Yes, we do. Thank you so much, Frank. It was excellent overview and a lot of information. So anyone who want to ask questions, this is time. We have still nearly 10 minutes or so. You can use microphone. That's actually easier for me to manage. Turn on your microphone, unmute from the lower left mute switch. Anyone? So there's one question in the chat line. I'm not 100% sure if I'm understanding, but it's about root knot resistance, root knot nematode resistance.
Yeah.
Asking guava root knot nematode. So that's...
Yeah, so the main gene that we've been using is the MI gene, and it doesn't confer resistance to all species. And it has limitations in that it doesn't always work under hot weather conditions. And, it looks like from some of the data we have that the genetic background that it functions in might affect, the efficacy of that gene. So I'm not totally up to speed on the available genetics for root knot. and also nematodes. You know, we have a lot of people on the team that specialize in that. That's on the tomato side. On the watermelon side, that's a great question. I don't know the mechanisms. I don't know that the genes have been mapped or they're known. That has not been shared to date.
Great. Anyone else ask questions? Don't be shy.
Microbiome, reading for microbiome, and
can you speak up? I can't hear
Yeah,
you.
so Dr. Panthi is asking the question about how can you breed for the microbiome? So that's a pie in the sky idea. But there's really good work on two parts. One in China, where they had rootstock that conferred resistance to verticillium. When they looked at the mechanism, they found that the rootstock had a different profile of exudates, and those exudates, normally a susceptible plant will cause verticillium microsclerotia to germinate. In the resistant rootstock, the microsclerotia didn't know the tomato was there, and so it was an avoidance mechanism. In other cases, if you think about plant pathology, just like, you know, we get specific genes that interact with specific microbes to give a specific effect, either resistance or susceptibility. Well, early work in Wisconsin, they could show that they could get specific genes that favored a specific microbe in the rhizoplane that suppressed a pathogen. So I believe as we learn more about the microbiome, as we learn more about genetics, tomatoes take up to 30% of their energy to exudate all kinds of stuff. And I believe as we understand metabolomics, genetics, we can breed for specific microbiomes. But that's a pie in the sky idea. The science is not there yet.
Interesting. So there's the other way around too, right? So the microbiome, so this particular genotype tend to create microbiome to attract pathogens. So it's sort of unintentionally creating that. So that's quite.
That's a great point. And maybe by default, we have been breeding for susceptibility in the microbiome.
Yeah.
Yeah. That's a good point, Jerry.
Yeah, because I have not a graftable crop, but I have one specific crop that tends to attract always, you know, pathogens, PCMs, and then I'm wondering if it is actually attracting somehow. But anyway, okay. Here's one question. Rootstock. What's the best rootstock used in Arizona area for watermelon summer production?
Yeah, you know, that's a question for Richard Hassel or Cherry Kubota or Mike Mayer, someone who works in Arizona. I didn't talk at all about melons. Cherry, you've done some great work.
Yeah, we did some work in Arizona, and summertime. Well, we always use the interspecific squash rootstock, and some of the rootstock was tolerant to the heat, and some of them are not so much. So I think it's important if you are looking at the fall planting. I think it's important to find a rootstock or check with the seed company regarding the tolerance to the heat. Winter time is likely have, you know, the spring planting usually have a big yield increase, and then we could get as much as 100% increase in our teeny tiny trial.
And Sherry, I think just like other countries, as a rootstock market matures, we'll breed rootstock that can tolerate cool weather, hot weather, salinity stress,
exactly.
Stress. You know, so as the market matures, I think we need a lot of on-farm research and support and collaborative work to, you know, test those questions.
Right.
But right now, I think the answers to those questions are available. You just got to find the person who knows it best.
That's right.
Yeah.
Anyone who want to ask using microphone, please go ahead.
Yeah, good morning, everyone.
Hi.
Yeah, I'm Fabrice Lisa from Cameroon.
Oh, welcome.
Welcome.
Yeah, I'm very grateful to participate on this conference. But I'm a young Cameroonian from the University of Yaoundé 1, and I have started, I'm writing a project on vegetable grafting. So I've grafted tomato on eggplants. So I wish to know if it is possible to share my project with you. And secondly, I wish to know if it is possible for a Cameroonian to benefit from the different PhD programs or the different PhD theses I've seen on this conference. Thank you.
Yeah, those a couple excellent questions. First of all, yeah, I think we have a lot of experts on the team that could interact with you. You know, there's teams in Florida that have done a fair amount of work with eggplants, and also at Clemson. And so there's a lot of data. There's a lot of data all over the world. In fact, the World Vegetable Center in Taiwan has some of the best expertise that I know of in Africa in terms of grafting. But we have people on our team that have spent a lot of time in different places. So yeah, so there's good expertise, and also there's a lot of opportunities for collaborative work. Research scholars have come for visiting for a short time. People have done PhDs in the U.S. and/or in their country, and so those have been worked out. The main thing is finding the source of money to do that work. The grant doesn't have that money, but
Okay, thank you.
yes.
All right. So there's another question in the chat line while some others want to talk. So what do you think about the use of grafting in the tropics according to the dynamics of the pathogens in this area?
Yeah, you would be a little bit like North Carolina, except a little bit worse. You have more aggressive populations of pathogens. The temperatures and the conditions favor high pathogen populations. So again, it's understanding what the pathogens are in the different fields. So there has to be a local knowledge about what the issues are for each field, farm, or area. And then I think you can choose really good rootstocks that will help you. So it is a combination of knowing the enemy and then choosing the right rootstocks.
Great. All right. Maybe one more question. Anyone?
Yeah, this is Carol, Washington State.
Hey, Carol.
And Frank, I'm wondering how much, Frank, is known about threshold values of the different pathogens. So at what level does it become economically profitable to use a grafted plant?
Yeah.
Information there is about that.
So there's, we've hit that directly and indirectly. So like for nematodes in North Carolina, we can sample soils in the fall. We know exact thresholds, and there's certain action thresholds that can be done. And that's not so much like if you have 80 nematodes per 500 cc's versus 120. It just gives you an estimate of the risk and whether or not you need to mitigate. And so there you might use grafting. So with nematodes, we have that. With verticillium, you guys have done great assays. Other labs can do great assays to assess verticillium inoculum. But overall, that data is not, there's not much of that data. And of course, there's so many things that affect level of disease, weather parameters, history of the land, etc. So it's hard to factor that in. So that's why we also did work on how do these rootstocks perform in the absence of disease. So if a grower feels like they have risk, they for sure don't want a rootstock that will decrease their yield. And what we've found is we can identify a rootstock that gives very good disease control where there's risk. And if there's no disease in certain sections of the field or in certain parts of the farm, they get equal or better yield than the control. Is that helpful, Carol?
Probably it's muted. Yeah.
So there's a lot of work to figure out how to do
Yes,
threshold.
yeah, sorry. There's a
Yeah.
delay in my clicking the mute button and it allowing me to talk, so I apologize.
Oh, no.
Yeah,
Yes,
no problem.
that's exactly what we're, trying to assess at what, and I realize it's very complicated.
Yeah.
So just curious what others have done.
That's a matter, there's a whole bunch of issues. We got really good diagnostic that can specifically identify the pathogen, but how many soil samples do you need to take to know your threshold's right? How do you know what the weather's going to be? So all these other parameters affect threshold determination.
Thank you.
Yeah.
All right. Anyone who wanna?
Hello.
Yes.
Hello, Dr. Kabuta.
Hi.
I'm Nawaz from Pakistan. Hello.
Hi, Nawaz.
I am Nawaz from Pakistan.
Yes.
Yes, we hear you
Thank
well.
you.
Excellent. Welcome.
I have. Thank you. I have one question. Can we use rootstock to control the airborne diseases such as powdery mildew? Here in Pakistan, we are facing a lot of problem related with powdery mildew on cucumber, on watermelon, and on muskmelon. Can you please guide?
Yes, I have.
Can you
I have
hear
a
me?
Yes.
Yes,
Hello?
we can hear you. And I have a twofold response. The first is the science is not mature enough, and there's not been enough work to say that rootstocks will confer powdery mildew resistance on a susceptible line of cucumber or watermelon. So that is not a current management strategy. The other part of that question is certain rootstocks are really susceptible to powdery mildew, and that becomes a problem when you're grafting. And so, there are breeding programs to get resistance in the rootstocks, or people have used biological control methods or other fungicides to control the powdery mildew on that rootstock. But I think that's a great question for the future. Can we breed rootstocks that affect the watermelon and cucumber lines to get less foliar disease? That's a very tough question to answer, but there's hope. There's some good literature there's some good work in the literature. There's a paper in China. There's also in citrus where they had rootstocks. It's a completely different crop, but it's really fun. Rootstocks conferred mite resistance. So there's some really good work out there, but no, there's no recommendations that we can give for rootstocks that give resistance against foliar problems.
It's great.
Okay, thank you.
You're welcome.
Thank you. Thank you.
That's quite interesting area too.
Yeah.
Silicon is somehow involved or something like that, no? Silicon
Yeah, that's right.
deposition in the leaves.
Yeah.
Yeah.
Silicon mediates
Right.
various defenses and
Right.
various mechanisms. Yeah.
Yeah. So that's quite interesting area. So I hate to say, but we already actually few minutes over. Well, I would like to thank again to Dr. Frank Louws, North Carolina State University, for excellent presentation. And I'd like to thank everyone in this space sharing the information and listening to the great info. And then hopefully you come back for the future webinar also. So thank you very much. Thank you.
Thank you. It's great to have everybody from so many different countries.
Thank you.
Mhm.